Enzymes. Enzymes are the most important group of proteins which are biologically active. They tremendously increase the efficiency of a biochemical reaction and are specific for each type of reaction. Without these enzymes, the reaction would proceed at a very slow speed, making the life impossible. Enzymes are composed of hundreds of amino acids joined together and coiled upon themselves to form a globular structure. The catalytic activity is restricted to a small portion of a structure known as active site. The reactant called substrate is attached to the active site, consisting of only a few amino acids, while rest of the bulk of amino acids maintain the globular structure of the enzyme. Some enzymes consist solely of proteins. Others have a non-protein part known as a cofactor, which is essential for proper functioning of the enzymes. The cofactor usually acts as a bridge between the enzyme and its substrate. Often it contributes directly to the chemical reactions, which bring about catalysis. Sometimes the cofactor provides a source of chemical energy, helps to derive reactions which would otherwise be difficult or impossible. Some enzymes use metal ions as cofactors, like magnesium, iron, zinc, copper, etc. The detachable cofactor is also known as activator if it is an inorganic ion. If the non-protein part is covalently bounded, it's known as prosthetic group. If it is loosely bounded to the protein part, it's known as coenzyme. It is closely related to vitamins, which represent the essential raw materials from which the coenzymes are made. Only small amounts of vitamins are needed because, like enzymes, coenzymes can be used again and again. An enzyme with its coenzyme or prosthetic group removed is designated as apoenzyme. Adding the correct concentrated coenzyme to the apoenzyme will restore the enzyme activity. An activated enzyme consisting of polypeptide chain and a cofactor is known as a holoenzyme. Many enzymes are simply dissolved in the cytoplasm. Other enzymes are tightly bound to certain subcellular organelles. They are produced by living cells for use in the near site of their production. The enzymes important in the photosynthesis are found in chloroplast, and enzymes involved in cellular respiration are found in mitochondria. Some of the enzymes which are involved in synthesis of proteins are integral part of the ribosomes. Characteristics of enzymes. Enzymes are the biochemical catalysts possessing the following important characteristics. All enzymes are globular proteins. They increase the rate of reaction without themselves being used up. Their presence do not affect the nature or properties of the end products. Small amounts of an enzyme can accelerate chemical reactions. They are very specific in their action. A single enzyme catalyzes only a single chemical reaction or a group of related reactions. They are sensitive to even a minor change in pH, temperature, and substrate concentration. Some enzymes require a cofactor for their proper functioning. They lower the activation energy of the reactions. Mechanism of enzyme action. Catalysis. An enzyme is a three-dimensional globular protein that has specific chemical composition due to its component amino acids and a specific shape. Every enzyme, by virtue of its specificity, recognizes and reacts with a special chemical substance called substrate. Any enzyme therefore reacts only with its specific substrate and transforms it into products. It's then released unaltered and thus can be used again and again. In certain cases, enzymes act in a series of chemical reactions in a particular order to complete a metabolic pathway, such as respiration or photosynthesis. The successive enzymes containing these reactions are normally present together in a precise order of reaction, such that substrate molecules can be literally handed on from one enzyme to another, forming an enzyme-to-enzyme chain. In this way, the products from one step in the pathway are transferred to the enzymes catalyzing the next step. An enzyme and its substrate react with each other through a definite charge-bearing site of an enzyme called active site. The charge and shape of the active site is formed by some amino acids present in polypeptide chains of the active site of the enzyme. These amino acids are brought closer and are arranged in a specific way by coiling and folding of the polypeptide chain within the globular symmetry of the enzyme. The active site of an enzyme is made up of two definite regions, the binding site and the catalytic site. The binding site helps the enzyme in the recognition and binding of a proper substrate to produce an enzyme substrate complex. This reaction activates the catalytic site. Activated catalytic site catalyzes the transformation of substrate into products. Thus, the enzyme after catalysis detaches from the products unchanged. Enzyme requires aqueous medium for its activity. Most enzymes do not flow about in a kind of cytoplasmic soup, but are attached to the membrane systems inside the cell in a specific and orderly arrangements. Mitochondria in chloroplasts are good examples of this. Emil Fischer in 1890 proposed a lock and key model to visualize substrate and enzyme interaction. According to this model, as one specific key can open only a specific lock, in same manner a specific enzyme can transform only one substrate into products. According to lock and key model, the active site is a rigid structure. There is no modification or flexibility in active site before, during, or after the enzyme action, and is used only as a template. Later studies did not support this model in all reactions. On the basis of new evidences, Koshland proposed its modified form in 1959. This is known as induced fit model. He argued that when a substrate combines with an enzyme, it induces changes in the enzyme structure. The change in structure enables the enzyme to perform its catalytic activity more effectively. Factors affecting the rate of enzyme action. The functional specificity of every enzyme is the consequence of its specific chemistry and configuration. Any factor that can alter the chemistry and shape of an enzyme can affect its rate of catalysis. Some of the important factors that can affect the rate of the enzyme actions are concentration of enzyme, concentration of substrate, temperature, and pH of the medium. Enzyme concentration. The rate of reaction depends directly on the amount of the enzyme present in a specific time at unlimited substrate concentration. If the amount of enzyme is increased by twofold, the reaction rate is doubled. By increasing the enzyme molecules, an increase in the number of active sites take place. More active sites will convert the substrate molecules into products in the given period of time. After a certain limiting concentration, the rate of reaction will no longer depend upon its increase. Substrate concentration. At low concentration of substrate, the reaction rate is directly proportional to the substrate available. If the enzyme concentration is kept constant and the amount of substrate is increased, a point is reached when a further increase in the substrate does not increase the rate of the reaction. This is because at higher substrate level, all the active sites of the enzymes are occupied. and further increase in the substrate does not increase the reaction rate. Temperature. The rate of enzyme-controlled reactions may increase with the increase in temperature, but up to a certain limit. All enzymes can work at their maximum rate at a specific temperature called optimum temperature. For enzymes of the human body, 37 degrees C is the optimum temperature. Heat provides activation energy, and therefore chemical reactions are accelerated at higher temperature. Heat also supplies kinetic energy to the reacting molecules, causing them to move rapidly. Thus, the reactants move more quickly and chances of their collisions with each other are increased. However, further increase in the heat energy also increases the vibrations of the atoms, which make up the enzyme molecule. If the vibrations become too violent, globular structure essential for the enzyme activity is lost and the enzyme is said to be denatured. pH value. Every enzyme functions more effectively at a narrow range of pH, known as optimum pH. A slight change in the pH can change the ionization of amino acids at the active site. Moreover, it may affect the ionization of the substrates. Under these change conditions, enzyme activity is either retarded or blocked completely. Extreme changes in pH causes the bonds in the enzyme break, resulting in the enzyme denaturation. Inhibitors. An inhibitor is a chemical substance which can react in place of a substrate, but is not transformed into products, and thus blocks the active site temporarily or permanently. For example, poisons like cyanide, antibiotics, antimetabolites, and some drugs. Inhibitors can be divided into two types: irreversible and reversible. Irreversible inhibitors. They check the reaction rate by occupying the active sites or destroying the globular structure. They occupy the active sites by forming covalent bonds, or they may physically block the active site. Reversible inhibitors. They form weak linkages with the enzyme. Their effect can be neutralized completely or partly by the increase in the concentration of the substrate. They are further divided into two major types: A. Competitive, B. Non-competitive. Competitive inhibitors. Because of the structural similarity with the substrate, they may be selected by the binding sites, but are not able to activate the catalytic sites. Thus, products are not formed. Non-competitive inhibitors. They form enzyme inhibitor complex at a point other than the active site. They alter the structure of the enzyme in such a way that even if the genuine substrate binds the active site, catalysis fails to take place.